This screencast is about active transport. So the last parts of the membrane function that we've covered have been passive transport, diffusion, simple diffusion, facilitated diffusion, and osmosis, another form of facilitated diffusion, but of water. So now we're going to be looking at active transport. So now we're going to be looking at how we use energy to move molecules against their concentration gradient. So remember, active transport requires energy, usually in the form of ATP, to move molecules against their concentration gradient. So you can see there's a low concentration here of these little ions, the squares that we're paying attention to here, and there's high concentration here. So for the cell to continue to keep moving more of these ions in, it takes some ATP, and there's this thing called a protein pump, and it's called a protein pump because it's made of protein, right? It's one of the integral proteins that is within the cell membrane. But you might also hear it referred to as an ion pump because it's pumping ions, or it might be named with the specific ion that it's pumping. Or sometimes you'll see it referred to as a proton pump because that's actually hydrogen ion itself. Okay, so in this case, we've got low concentration here, high concentration here. ATP is added to this to this ion pump, and then it's going to move the ions into the cell against their concentration gradient. The way that ATP works actually, so here is what ATP looks like. It actually looks like an adenine nucleotide, but it's got two extra phosphates on the end. And when the energy that it's going to be giving is in this bond here between the two phosphates, the last two phosphates. And so this phosphate will often be donated to the molecule that it is giving energy to. And so you'll see there'll often be a temporary bond with an extra little phosphate there, and that's got the energy. And now the ADP is in its low energy state and needs to be regenerated to ATP through cell respiration. All right, so we looked at using ATP to just pump ions across, but sometimes there's also a way that active transport happens using something called a co-transporter. So a co-transporter is when we use one molecule that's moving along its concentration gradient to provide the energy for a second molecule to move against its concentration gradient. So in this case, energy gets used by this proton pump to move a bunch of hydrogen ions outside the cell. So now the concentration of hydrogen ions are high here and low here. So now it's going to want to diffuse down along its concentration gradient, and it can through this co-transporter. And the sucrose is actually going to be in lower concentration here. It wants to keep a high concentration so there's plenty within the cell. So for that sucrose to move against its concentration gradient, it uses the energy of the hydrogens diffusing through the co-transporter, and then it can move that sucrose across. I'm going to show you a little video example. Okay, so in this little video example, you're going to watch this video in totality, but what it's showing you here is this is the small intestines, and this is inside the gut, so where all the food and yummy things that you want to bring into the cells of the intestine. So we want to move it from here into the cells so your body can then transport it in the blood to other places. And there's a couple of examples. There's tons of different protein channels. There's facilitated diffusion happening. There's osmosis happening. You'll see when you watch the video. But I'm just going to focus on, here's a quick example of an ion pump. This is a sodium-potassium pump. We're going to learn this in detail in another video. And you'll see it's going to be moving sodium in one direction and potassium in the other. So right now, the sodium potassium pump is open here to the inside of the gut, and 3 sodium ions are going to be coming into the open. And you see this thing? That's an ATP. So that ATP is going to come and it's going to leave a phosphate. So there's that phosphate I was talking about. And that allows the pump to change shape. And then the sodiums are going to leave. And then you're going to have a couple of potassium ions come in, and those will then be dropped off inside the cell. We'll look at this type of pump more closely in another video, but realize we used energy to move ions against their concentration gradient. Here's an example of co-transport. So all those sodium ions that we pumped out, those little yellow ones, So the sodium ions that we pumped out, we're going to use those in co-transport in the gut to bring in a sugar molecule. I think in this case, it might be glucose. And so you can see let me come back to that. Okay, so in this case, the sodium ions are going to diffuse down along their concentration gradient, and then the glucose molecule will also be allowed to be moved against its concentration gradient. So you can watch, they both come in together. And the sodium ions got the high concentration here because of that sodium-potassium pump that we looked at before. All right. Now, the next question we want to look, we are going to now focus on endocytosis and exocytosis. So that is bringing things into the cell and out of the cell using vesicles. So here is an example of exocytosis. I think you remember we were looking at when proteins get made in the endoplasmic reticulum, they get transported in vesicles to the Golgi, which they're processed, and then they'll be secreted. So if this were, say, a pancreas cell and it was making insulin, it would make the insulin here at the ribosomes on the rough endoplasmic reticulum, send them in vesicles to the Golgi, where they get modified and packaged up into another vesicle, which then get exported out of the cell because the insulin needs to enter your bloodstream. So then it will be, you can see this is a vesicle that has a plasma membrane, and when it gets to the edge, that plasma membrane will fuse with the membrane and spill the contents out. That is called exocytosis. It's exiting the cell. Endocytosis is when the cell is actually taking in something like food or liquid, and the plasma membrane, because it's fluid, can actually wrap itself around what it wants to bring in. It will engulf it, and then it'll have a food vacuole, which it will often end up, kind of digesting. So if you remember when we were looking at phagocytosis with the white blood cells, with those phagocytes, that's what they were doing to the viruses and different pathogens that they would take in. They would take it in, engulf it through endocytosis, and you can also, when it's eating, you can call that phagocytosis. They would bring it in through phagocytosis, and then there would be molecules that would then digest it and be able to present the antigens. Okay, so you can see phagocytosis, that means cell eating, but it's bringing it into the cell is endocytosis.